right, welcome to 3.5 part 2, almost done here. All right, in our last lecture, we talked about, we reviewed PCR, we looked at gel electrophoresis and using it for paternity and crime scenes, and analyzing DNA profiles using that method of either the proteins being through the gel or the DNA. We looked at all those different ways in which gene transfer occurs, and in class we're looking at ethics. So in this one we are just looking at cloning, right, which is a lot, but we're going to define what cloning is, some natural and artificial cloning methods, and again a little bit of the ethics of it. Okay, so let me go back some, right, preview, there we go. Okay, GMOs. Looked at this in, excuse me, in class. And pollen, here we go. All right, cloning. So I think most people have a pretty good idea of what cloning is, but let's talk about like scientific definitions and then the uses of it. So clones, right? The actual noun here. Oh, that was a wonderful graphic. Are groups of genetically identical organisms derived from a single parent cell. So I have a single parent cell and then genetically identical organisms that are created from it. Cloning can occur naturally, right? So in asexual reproduction, when I get just exact copies made over and over, those are natural clones. It can also occur artificially in ways kind of like embryonic division or somatic cell nuclear transfer when I'm creating exact cells from a parent cell, right? So artificial and natural ways there. Okay, so here's some different natural ways that we talk about cloning in animals, right? So certain animal species clone themselves in a variety of ways. You might already know about these things like starfish, right? Maybe not the planaria or the hydra, but let's start here with fragmentation because that is probably the most familiar. So fragmentation is when the animal is able to clone itself in fragments. So if you cut a starfish up, right, or the arm off, it'll actually create an exact copy, a clone of that starfish, right? It's able to regrow. That's called fragmentation, right? So cut off the arm here, able to grow into this one and this one being identical, okay? Hydras, maybe a little less familiar with those. Clone by a process called budding, which is when the cell splits off from the parent, right? Or cell split off from parent organism, right? So I've got this parent organism here. It's got both of these. This little kind of hydra-like arm splits off, and now I have two genetically identical hydras, right? Another example is planaria, which is the organism divides equally into, we've already talked about this a little bit, binary fission, right? So binary fission, right? The process we're moving apart, binary meaning two, and it's a way in which cells divide. In this case, we're looking at planaria, right? Dividing into two identical. Okay, so this is in animals. In plants, again, some things that you're probably more familiar with. So plants, what we call cloning in there, they have the capacity of what's called vegetative propagation, which is where small plants can be induced to grow independently. So if anyone has parents that are into gardening or anything like that, where they're able to actually cut off the bud and the node, and those plants retain their pluripotent tissues, the meristem, the stem cells, right? So in stem cutting, they're separated portion of plant stems that can regrow into new independent clones. So here's a stem, right, possesses what are called nodes from which leaves, roots, or branch grow, right? So here's a node here. Here's its budding off. And the various factors may affect the rooting or the stem cutting, right? But I can actually take this little area and I'll grow a root and a leaf from that little area, right? The nodes, because it has all that stuff. I'm trying to remember where I was just reading about this. I think it was rose bushes. Like you're able to take, like, this is like, from prized rose bush, and you're able to take something that genetically is really, whether it's prized or with crops being really resistant to something, so you're actually able to clone that certain crop, right, or vegetable or plant or whatever it is, and take it and have the same exact DNA, right? So it has those same properties. Okay, so natural cloning. All right, humans, right? The only natural cloning that we know about are identical twins, right? So humans, what happens when identical twins, we talked a little about, they're called monozygotic versus dizygotic. This is when I have a single cell, so single sperm and egg is fertilized, so I have exact DNA, and before it even has a chance to start developing, it splits in two. So it takes that DNA and splits in two to make two identical cells that will now have the same DNA, same directions, and create two identical human beings genetically, right? If you know some identical twins, you know that they still look different ways. We'll talk about why that might occur, right? If they have the same DNA, different genes are expressed. But this method of cloning can also be used artificially, right? Know that this separation must occur early, which is the Marola stage. And this cloning method is limited by the fact that it still occurs after sexual reproduction has occurred, right? So it has to happen really early after the sperm meets egg, and it's very hard to catch it in that state, right? In the Marola stage before it has started to reproduce into two cells, right? And so this can be done artificially, but it's limited. Okay. All right. Now let's talk about artificial cloning, and we'll be done. So artificial cloning. This is when we get into like the ethical parts of cloning, but we have seen in different countries in different ways clones that are developed artificially using different. So we'll talk about two things. So tissue, right, and then whole animals. We'll talk about that in a second. So if I'm just using differentiated adult tissue, right, when we looked at stem cells a long time ago, we call this method, so I have the differentiated adult tissue, and we can clone that using what's called somatic cell nuclear transfer. So somatic cell, right, so being body cells, nuclear, all that DNA, transfer, SCNT. All right. So why would we do those two things, right? Two main purposes. Therapeutic is probably the one that you are most familiar with. So therapeutic cloning produces new tissues, right? So be able to grow skin cells outside of the body, be able to replace or recreate different places that have been damaged, right? Transplants, disease treatments, giving bodies the new cells that they need if they have any damage in the ones that exist, right? So this is called therapeutic cloning. Reproductive cloning is when I'm taking the entire new organism. So reproductive cloning is used in places like repopulating endangered species. So endangered species that have happened because of, or have gone endangered because of humans. There has been, what I want to say, they're able to take those cells and then recreate, whether it's implanting in the womb, right, of an animal that isn't pregnant already, but it's trying to repopulate endangered species. Therapeutic is just the tissues, right? Currently, this technique, for a couple different reasons, has low success rate when applied to reproduction. So talking about humans, human reproductive cloning is considered unethical and is currently illegal, right? So we do not see any of the complete reproductive cloning in humans, but we do see the therapeutic cloning used for treating diseases, transplants, such like that. Okay, here's a little bit of that process of the SCNT. I promise this is the second last one, right? So because you had to know how the plasmids and the vectors and all that worked in the last one, you do need to know the process of artificial cloning. Use it, and then think about that though, right? The somatic cell nuclear transfer, right? Taking that nuclear and then implanting it. It has all the directions into a new cell, right? So here's how it happens. So the nucleus is removed from an adult body cell, right? The egg cell nucleus is destroyed by UV radiation. So enucleated egg here, right? And then the body cell nucleus fuses to the enucleated egg, and then electrical current stimulates the egg to divide, right? Develops an embryo implanted into a surrogate. This is what I was talking about with the endangered, right? And the embryo develops into a cloned animal. So body cell extracted, enucleated egg, removing the nucleus, fusing the nucleus into a new egg, right? So I kind of skipped over this. This nucleus is removed. The egg cell, from an egg cell like that would be like from that animal, right? That nucleus is destroyed by UV radiation. So removing whatever this one is. And that body cell nucleus is then put into the enucleated, so the egg that does not have a nucleus, and then is stimulated to divide. So, and then that embryo, right, just like, in vitro fertilization, right, is then put into the animal that is endangered or whatever we are trying to repopulate. Okay, so SCNT. Similar, I guess, kind of the last one where I'm taking the portion that I want, putting in, but this one's taking the entire nucleus and putting it into an egg so that it can develop into a cloned animal. All right, that's it. There it is. We're done. Okay. We'll talk more about, like I said, ethics in class, but we're done with topic 3. Only one more left, folks. Have a wonderful day, and hopefully this is the last thing you're doing before break. I'll see you guys soon.